Okay, welcome to the last podcast series of the year. It's on photosynthesis. Let's get to it. We're going to do this in three parts. We're going to do an intro to photosynthesis and basic things about leaf structure, chloroplast structure, things like that. Then we're going to look at the two sets of reactions required, to make photosynthesis work. Okay? You are looking at a lot of photosynthesis going on right there. You're seeing a lot of chlorophyll. Chlorophyll is the key molecule that allows photosynthesis to occur. It is the key that takes that ultimate energy source of sunlight, the UV radiation that comes from that, and converts it. The chlorophyll converts that to a form that living things can use, a chemical form. Living things aren't very good at using UV light. We have to convert that into a chemical form of energy that we can readily use. Okay? So, here we go. Light, air, and water, the things we need to do photosynthesis. All of life, in order to keep going, we are fighting entropy. Our bodies want to fall apart. When we can no longer fight that, we're dead. Okay? So we are fighting entropy all the time, and we need a constant input of energy to do that. Okay? Now, heterotrophs, like you and me, and autotrophs, like plants, both need this input of energy. Okay? Both of us get this input of energy from food. The energy we get is both of us from food. Okay? The key difference is that as consumers, heterotrophs eat this food. They take it in from the environment, they eat it, already made. Autotrophs, however, make their own food. They produce their own food, hence the ecological phrase producers. Please do remember the difference between consumers and producers and their ecological roles. That will certainly come up on the AP exam. So, the way that the autotrophs we're going to be talking about in this podcast make their own food is by photosynthesis, of course, and that is by converting the energy of sunlight into food. Okay? Now, they don't just magically take sunlight and poof, there's food. Sunlight is just the energy. If you're going to build a Lego tower, which we need to think of a molecule of glucose as being a Lego tower. It's a big complex molecule. To put that together, you need energy to do that. Well, plants need energy to build glucose. Sunlight is the energy used to do that. Okay? So, obviously that's called photosynthesis. So, heterotrophs and autotrophs both need food. Okay? It's just that we get it in different ways. Autotrophs make their own, heterotrophs have to grab it from the environment. Alright? Now, what we both share totally in common is that once we have food, we have to convert those food molecules, those glucose molecules, into a usable energy form by way of cell respiration. So essentially we have to convert this food to usable energy, which we normally refer to as ATP. And both of us have to do that. Animals, consumers have to do that. Plants have to do that. Okay? It's just that we can't make our own food. They can. They have chloroplasts, organelles that make food for them. We don't have those. We do have mitochondria that break down food for us and get ATP, but they have mitochondria too to break down the food they make to get ATP. Okay? So, make sure you understand that. A lot of kids come away thinking animals have mitochondria and plants have chloroplasts. No. Animals have mitochondria and plants have mitochondria and chloroplasts. Okay, so let's make sure we've got that. Now, heterotrophs and autotrophs have to do this process. They don't necessarily both ingest their organic molecules, but they do have organic molecules that they obtain one way or another and have to break them down. This is cell respiration. This is the equation we learned back when we did glycolysis and Krebs and the electron transport chain. You take in glucose, you take in oxygen, and you do some chemical reactions and end up with CO2, water, and hopefully usable energy in the form of ATP. And this is the way it is correctly written chemically. Okay? Now, autotrophs do this. Okay? I'm sitting here pointing at the screen like you can see what I'm pointing at. They do this in their own mitochondria. But this glucose that they break down is something that they made in this process we're going to look at right here, photosynthesis. Okay, CO2 plus water plus light. The light energy is the energy used to kind of force these atoms together, in a very simplistic way of thinking of it, into that glucose molecule, that big Lego tower. And then we get O2 as a waste product that fortunately fills the atmosphere with air that we can breathe so that we can do this process up here. Notice we need this O2 up here to do this process. The cell respiration is an exergonic process. Energy is released in the form of ATP. This product and this product have less energy in them than the reactant did because we created this energy form over here, and so all this energy is going to go off and be used. Here, this is an endergonic process, meaning these reactants here and here have far less energy in them than this than this product does, and that's because this light energy was put into here to fuse these guys together to create that. This is an oxidation reaction. This is an oxidation. We are going to break down and strip electrons from glucose here. This is a reduction reaction, this endergonic reaction. We're going to add electrons to CO2, and we're going to end up getting a complex carbohydrate, that glucose. Okay? All right. Plants, please remember, are not the only autotrophs. They're not the only photosynthesizers. Bacteria can do photosynthesis. They don't have chloroplasts because they don't have organelles, but they do have chlorophyll, the chemical that does photosynthesis. So cyanobacteria are a photosynthetic bacteria. It is thought that they contributed greatly to putting enough oxygen in the atmosphere to support life, aerobic life forms like us. Protists, there are many algae that are also photosynthetic, like this sea lettuce here. So it's not a plant, it's green like a plant, but it isn't a plant. Again, they can do photosynthesis too. Okay? Now, excuse me, if you are a plant, you need to be able to take light and you need to be able to convert that light into a form of energy that is more stable than light. But remember, ATP is still not terribly stable. It gets used up pretty quick. But when we have this ATP, we are going to use that to convert into an even more stable form of energy. So we're going to take light initially, convert it to this, but ultimately we're going to convert the ATP into glucose, which is a much more stable form of energy. I want you to kind of keep this idea in mind because photosynthesis is really two sets of chemical reactions. The first set we're going to learn about is this conversion of light to ATP, kind of a temporary storage form of energy. The second set of reactions is going to take the ATP and convert it to glucose, which is a little bit more permanent, though still readily used. Okay? Now, we are going to need some raw materials to create this glucose molecule. The raw materials we need are water and CO2. Of course, plants need other things too. They need minerals from the soil and things like that, but we are focusing exclusively on photosynthesis now, so these are the only raw materials that we're going to talk about besides light. Okay? All right. Now, to get these raw materials, plants have very unique bodies. We've already talked about that. They have a top half, if you will, and a bottom half that are designed to do two different things. The top half is designed with the broad green leaves to receive sunlight. They're like little solar collectors. The leaves also take in CO2 from the atmosphere. The stomata are the holes in the undersurface of the leaf that allow CO2 to come into the leaf. Water is also obtained, but not from the top half. That is obtained from the bottom half that has got naked tissue there with no waxy water resistant covering, because we need to be able to absorb water from the soil, and so osmosis will be occurring to take water into the root, and so on. Okay, we're also going to get nutrients from those same roots. When we say nutrients, we mean things that are not like sugar, nitrogen, phosphorus, potassium, sulfur, magnesium. This is really important because this is at the heart of chlorophyll molecules, iron even, so on. So things like that are going to be taken in with the water in the roots. All right, you need to have a good understanding of leaf structure. Okay, so let's remember how a leaf, I know you were taught this earlier in your school career. So the top is actually the tippy top here is cuticle, which is made of wax and prevents water loss, water evaporation from the surface of the leaf. We then have a layer of single cells, that's epidermis, which is for protection. The mesophyll, which is all of this in the middle, is where the photosynthesis happens. There are two layers to the mesophyll. The top part with these cells here is called the palisade layer, and that is where most of the photosynthesis happens. There would be full of chloroplasts in these cells here. Okay? Down here we have what is called the spongy layer. As you can see, it's got lots of spaces in it. This is where the gas exchange occurs. So this layer, the palisade, is the photosynthetic layer. This layer is for gas exchange. The bottom layer is simply the lower epidermis, and that's where we find the stomata, the stoma, whatever you wish to call them, for gas exchange. Okay, remember we lose water vapor from here as well as taking CO2 in, so that's kind of a double-edged sword. So palisade layer, photosynthesis, spongy layer, gas exchange, lower epidermis. There's an excellent picture of a stoma or stomata. They go by all kinds of different variations on that, but that's the hole surrounded by the guard cells that will open and close to let water pass or not pass, and so on. Okay, basically we've just talked about all that, so I'm skipping it. Do remember the role of the stomata in controlling transpiration, because if we're in a time of drought, losing water through these things is not good. But if we close them, that means we don't get any CO2 in, and that's not good either, because then we can't do photosynthesis. So this is kind of a double-edged sword here. We have to, it's a means of control. It's not a perfect solution. All right, now what we need to do is go into the leaf and go into where the action is happening. We need to understand the structure of a chloroplast. Please remember that chloroplasts and mitochondria are believed to have a similar origin, and that they both were originally free living bacteria that were consumed by a larger cell, but instead of being eaten, they began engaging in a symbiotic, mutualistic, symbiotic relationship. Mutualistic, symbiotic relationship, meaning something that was a benefit to them and something that was of benefit to the cell and which had engulfed them. Remember that this is called endosymbiotic theory, and we have lots of evidence to support it, not the least of which is that chloroplasts and mitochondria both have their own DNA molecules. They both can reproduce within the cells in which they exist, and many other qualities that suggest this is true. Okay. All right. So what we're doing is we're going down into the leaf, and we're going into one of these palisade cells, and we're finding a chloroplast in there. So that is a chloroplast inside a palisade cell, and this is that chloroplast blown up in diagram form. And so we're going to look at it over here. The chloroplast, the other evidence, that's what I was trying to remember, is that they are both double membrane structures, the mitochondria and the chloroplast. The inner membrane was probably derived originally from the bacteria that got consumed. This was probably derived from the host cell. Now, inside this chloroplast, we have structures that contain chlorophyll, okay? And that's where the action is. So let's see, alright, this is what we want to do. We want to talk about the structure of the chloroplast, okay? Double membrane, I already mentioned that, inner, outer. Now, the stroma is a liquid-filled structure. Okay, if this is my chloroplast, it's the liquid-filled area in the chloroplast. So all of this would be stroma, okay, fluid-filled interior of the chloroplast. Now, what breaks up the stroma are little membrane-bound structures inside the chloroplast that are called thylakoids. This is where the action happens. The thylakoid membranes are the sites of photosynthesis, and we see that they're all kind of connected, but they look like stacks of coins inside the chloroplast, okay? And they are surrounded by the stroma. A granum is a stack of these thylakoids, so that's a granum. And the thylakoid membrane, the membranes of these little oval structures here, of these little coins here, are where the chlorophyll molecules that catch the energy of sunlight reside. Okay? That's where the light trapping action happens. And without that, nothing else can happen after it. Alright? It's important that we understand what a chlorophyll molecule looks like, A, because you might see one, B, it is a striking similarity to another pigment molecule that you might be familiar with, and I thought I had included a picture, but clearly not. Hemoglobin looks almost exactly like this, but in the middle, instead of magnesium in a hemoglobin molecule, you've got iron, okay? Hemoglobin being what makes your blood carry oxygen. Well, chlorophyll has magnesium in the middle, and this gives it the green color that we see, this green pigment. This atom is very hard for plants to come by. It's not cheap to make chlorophyll for a plant, and so that's why in the fall, rather than just allowing the green leaves to fall to the ground and wasting all this precious magnesium, the plants resorb the chlorophyll, resorb the magnesium, and that is when we begin to see the yellows and the oranges and the reds of the autumn leaves, because the plant resorbs all that chlorophyll so it can reuse the magnesium in the next year. There are other pigments. They are not the photosynthetic pigment that chlorophyll is, but that's what we're seeing in the fall, are those yellows and reds and so on. Okay. Now, before we jump into our reactions, we're going to wrap this up with just trying to make sure we understand how light behaves. Light can be both a wave and a particle. We are going to be talking about it acting as a particle, as a photon, a fixed quantity of energy. Okay? Now, chlorophyll is really good at absorbing photons in the red and blue parts of the visible light spectrum. Okay? It's not so great at absorbing other wavelengths of light, other kinds of photons. Okay? What chlorophyll definitely does not do is it definitely does not absorb green light. That is why when we look at chlorophyll, we see green, because that green light is not being absorbed by the plant's leaf, it is being reflected by the plant's leaf. It's not being absorbed, okay? So red and blue are being absorbed, okay? And you can see, if you look at what wavelengths are being absorbed, you can see the peak in the blue and the peak in the red end, and a very, very low section here in the green, showing that this is being absorbed and these colors are being absorbed, these colors not so much, okay? Now, there are several types of chlorophyll. You should have an understanding of them and other pigments. Chlorophyll A is the main photosynthetic pigment. It is the one that is actually catching the light energy and converting it to a usable chemical form. Okay? Looking like that. Directly participates in the photosynthetic reactions. It is a blue-green in color. If you pull it out of the plant and look at it, we did a chromatography lab that some people were here for. There are also accessory pigments that help chlorophyll A do its job and help the plant in other ways. They catch light from the sun and then pass it to chlorophyll A so that chlorophyll A can do its magic with it. One of these is called chlorophyll B. Okay, it is an olive green color, more or less. Maybe kind of like that, maybe a little bit more olivey. There are also pigments called carotenoids that are the oranges and the reds and some brilliant yellows as well. Now, these carotenoids are often serving as photoprotection, and what that means is they are going to protect delicate tissues in the plant from UV radiation. So the accessory pigments have two roles: to pass light energy to chlorophyll A and to protect the plant from UV radiation. Okay? Be aware of what the visible light spectrum is, and that chlorophyll is pretty good at absorbing here and down in here, and not so good in the middle. Okay, Roy G. Biv and all that. Okay, chlorophyll A, we already talked about this, we already talked about that, we already talked about that. Carotenoids, xanthophylls are another type of yellowy, orangey, reddy pigment, so just so if you ever see that, these are all just accessory pigments. Okay? Alright, now, when a molecule absorbs a photon, a light particle, so to speak, a piece of light, electrons get elevated to higher energy levels, and thus if an electron is elevated to a higher energy level, it has more potential energy that could potentially be utilized. The normal orbital, where the electron normally lives, is ground state. The higher orbital is called the excited state. Okay, so light energy causes the electron to move to an excited state. Okay, chlorophyll is really, really good at absorbing photons at this wavelength, 680 nanometers and 700 nanometers. Okay, now, normally, once we have excited electron, it just falls back down to its normal orbit, to its ground state. But the thing about chlorophyll is that it can get excited, an electron can get excited, and there is another atom, a molecule nearby that will catch and hold that electron before it gets a chance to fall back down and rejoin the chlorophyll molecule. And this is the key part of trapping the energy of sunlight into a chemical form. We're going to catch electrons at a higher energy state and not let them fall back to a lower energy state. Alright, that's it for this part. We are then going to fall into doing the photosynthetic reactions. Alright, thanks for watching. I'll catch you in the next part of this podcast.